This is a recording of the journal article titled Human-mediated and natural dispersal of an invasive fish in the eastern Great Lakes, authored by Johansson, Bradley Dufour, Kyle Wellband, Linda Corkum, Hugh MacIsaac, and Daniel Heath. Published in 2018. Abstract. The globally invasive round goby Neogobius melanostomus was introduced to the Great Lakes around 1990, spreading widely and becoming the dominant benthic fish in many areas. The speed and scope of this invasion is remarkable and calls into question conventional secondary spread models and scenarios. We utilized 9 microsatellites to identify large-scale genetic structure in round goby populations in the eastern Great Lakes and assessed the role of colonization versus secondary transport and dispersal in developing this structure. We identified 3 clusters corresponding with Lake Huron, eastern Lake Erie, and western Lake Erie plus eastern Lake Ontario, along with 3 highly divergent populations. Bottleneck analysis identified founder effects in 2 divergent populations. Regression analyses of isolation by distance and allelic richness versus distance from the initial invasion site were consistent with limited migration. However, some populations in eastern Lake Erie and Lake Ontario showed anomalously low genetic distance from the original site of colonization, consistent with secondary transport of large numbers of individuals via ballast water. We conclude that genetic structure of round goby in the Great Lakes principally resulted from long-distance secondary transport via ballast water with additional movement of individuals via bait buckets and natural dispersal. The success of round gobies represents an interesting model for colonization characterization. However, those same attributes present significant challenges for conservation and fisheries management. Current management likely prevents many new species from arriving in the Great Lakes, but fails to address the transport of species within the lakes after they arrive. This is an issue of clear and pressing importance. This is the end of the abstract. Introduction. Of all of the human impacts on global biodiversity, biological invasions are perhaps the most damaging, worst controlled, and most difficult to mitigate. Although species have been colonizing new areas for much of geological time, the current rate of species introductions and seemingly idiosyncratic biogeographic nature would seem to be unprecedented in global history. Existing evidence suggests that freshwater ecosystems are heavily invaded and that hundreds or thousands of species have been introduced to freshwater ecosystems worldwide, including such damaging invasives as the floating water hyacinth Eichhornia crucipes, Asian clam Corbicula fluminata, and spiny water flea Bithotrephes longimanus. Organisms are transported and released via a number of major vectors, including intentional stocking, escapes or releases from aquaria, gardens, or bait buckets, escapes from aquaculture and horticulture, transport in ballast water, and dispersal via man-made canals. Most of these vectors are selective in terms of what taxa they transport, and invasions in general are taxonomically non-random. Because of their economic food and sport value, fishes such as common carp Cyprinus carpio, various salmonids, family Salmonidae, and European perch Perca fluviatilis, among others, have been intentionally transported and stocked worldwide. Other species such as the western mosquito fish Gambusia affinis were introduced for biocontrol purposes or introduced unintentionally, as in the case of the red lionfish, Pterois volitans, in the Caribbean Sea. As a result, fishes are among the most introduced organisms globally, i.e. 430 out of 744 and 153 out of 432 established alien species in fresh waters of North America and Europe, respectively. The round goby, Neogobius melanostomus palus, is a relatively small, less than 25 centimeters in maximum length, benthic fish species native to the Ponto-Caspian region of Eastern Europe. Now listed as one of the 100 worst invasive species globally, the round goby was first found in the Laurentian Great Lakes in 1990 and in the Baltic Sea in 1991. It subsequently spread to all 5 Great Lakes and is continuing to expand its non-native range in both North America and Europe. Specific traits that provide the round goby an invasive advantage include its broad tolerance of environmental conditions, diverse diet, high fecundity coupled with paternal offspring care, and aggressive behavior in acquiring food and spawning sites. The species has become the dominant benthic fish in many areas of the Great Lakes and is of great concern owing to intense predation of native fish species' eggs. Development of canals and waterways and the increase of commercial shipping and recreational boat traffic across both Europe and North America have provided new dispersal pathways and opportunities, greatly contributing to the species' rapid spread. Previous research has demonstrated that Great Lakes populations of round goby are characterized by significant population structure, and that this population structure has remained relatively consistent over time. This structure has been suggested to result from a combination of factors, including variation in the population subsamples originally introduced to different areas, natural dispersal of fishes, and human-mediated dispersal via ballast water and bait bucket or other small-scale transport. However, none of these mechanisms has been tested in a rigorous framework at the large scale. Each of these introduction or transport mechanisms should leave a characteristic genetic signal in the population and is discussed in detail below. Natural dispersal of the round goby was previously thought to be limited. Adults display high site fidelity with home ranges conservatively estimated at 5 plus or minus 1.2 meters squared. But individuals occasionally migrate much greater distances, i.e. up to 2 kilometers. In addition, while larvae exhibit diel vertical migration, the consequence to natural dispersal is likely small since the larvae are negatively buoyant and remain near the surface for only very limited periods. Paradoxically, the supposed tendency for limited movement in round goby is juxtaposed by the rapid spread to new sites. For example, in tributaries on the Great Lakes, upstream spread has been estimated to range from 0.5 to 4 kilometers per year to the exponent -1, while in estuarian systems it can be an order of magnitude higher, i.e. 30 to 50 kilometers per year to the exponent -1. Spread may be facilitated in part by intraspecific competition or aggression, which could promote migration of subdominant individuals out of areas of high density. Gradual natural dispersal of small numbers of individuals, as would be expected of round gobies, should lead to an identifiable pattern of isolation by distance, where populations become less and less genetically similar with increasing geographic distance. While natural dispersal has been suggested to play some role in the population genetic structure of round gobies, by itself it is clearly insufficient to explain the population genetic patterns previously identified. In addition to natural dispersal, freshwater ballast water transfer moves round gobies within the Great Lakes ecosystem. In this case, nocturnal ballasting operations that correspond with the nocturnal pelagic feeding of larval round gobies could potentially take up thousands of individuals and subsequently transport them to seed new areas or add novel genotypes to established populations. This scenario is consistent with the rapid and substantial range expansions observed throughout the Great Lakes, and shipping has been demonstrated to be negatively related to pairwise genetic differentiation in goby populations in Lake Michigan. Transport via shipping within the Great Lakes system could lead to a pattern of anomalous similarity between geographically disjunct and potentially distant populations, or may function to diminish any isolation by distance signal in the genetic data. Transport and ballast within the Great Lakes is also consistent with previous genetic research that suggested that some outlying, relative to the original invasion sites, show high genetic diversity. The once common practice of using round gobies as bait may also have played a role in their dispersal, as they have been found in areas where no ballast water is discharged. While bait bucket transfers between large, established populations is possible and would serve as a source of migrants, possibly with little overall impact on the population genetic signal, populations established via bait bucket transfer should show evidence of founder effects and would be characterized by low genetic diversity and could be highly distinct due to the random sampling process. Earlier studies have demonstrated that introduction, dispersal, and consequent range expansion of the species in the Great Lakes is likely a complex process characterized by multiple modes involving both natural dispersal and human assistance. However, those mechanisms should result in characteristic population genetic signatures, and hence some understanding of the round goby colonization of the Great Lakes should be possible through detailed genetic analyses. The goals of this project were twofold. First, we identified large-scale genetic structure in round goby populations in and around Ontario, Canada, and second, we tested specific hypotheses about the role of initial colonization versus secondary transport and/or natural dispersal in building this genetic structure. We predict that the cluster around the original founding population in Lake St. Clair will have the highest genetic diversity, and that diversity will decline with distance from Lake St. Clair. Because shipping within the Great Lakes moves both huge quantities of ballast water annually and huge numbers of individuals simultaneously, we expect that ballast water transport may play a dominant role in the population structure of round goby in the Great Lakes. Thus, we predict that distant populations may be anomalously similar if located near major ports or along shipping routes, and should show little or no decrease in diversity with distance, while populations away from shipping routes should show clear patterns of founder effects. In addition, we predict that diffusive dispersal will be the principal mechanism that fills in areas away from deballasting locations, so that evidence of isolation by distance should be apparent with increasing distance from such points. We test each of these hypotheses using a suite of population genetic tools at both the population sampling location and individual level. This is the end of the introduction. Materials and methods. Sample collection and geographical information. Round gobies, total N equals 1,958, were sampled from 32 sites in Lakes Huron, Erie, and Ontario in summer and autumn of 2005 and 2006. Sampling sites were selected based on known presence of the species, accessibility, and ease of capturing sufficient sample sizes. The number of fish collected per site ranged from 21 to 128, with an average of 61 individuals per site. Fish were collected using a variety of sampling techniques, including seine nets, hook and line, and benthic trawl or hand spearing, depending on sampling site. Fish were humanely euthanized in accordance with Ontario law and University of Windsor Animal Care Protocol. A small portion of the caudal fin was collected and stored in 95% ethanol. This is the end of the sample collection and geographical information section. Microsatellite genotyping. DNA was extracted using a Wizard genomic DNA purification kit, or the 96-well glass fiber plate protocol described by Elphinstone et al. In 2003. All samples were genotyped at 9 polymorphic microsatellite loci following Dufour et al. In 2007. PCRs were prepared in 7 microliter total volumes containing 50 to 100 nanograms of template DNA, 1x PCR buffer, locus-specific concentrations of magnesium chloride, 0.2 millimoles of each DNTP, 0.025 micromoles of IR dye, infrared dye labeled forward primer, with 0.05 units of Taq DNA polymerase. Thermocycler profiles consisted of a 2-minute initial denaturation at 94 degrees Celsius, followed by 35 cycles of 15 seconds at 94 degrees Celsius, 15 seconds of locus-specific annealing temperatures, 30-second extension at 72 degrees Celsius, with a final extension step at 72 degrees Celsius for 2 minutes. Amplified PCR products were visualized on a LICOR 4300 DNA analysis system. Allele sizes were determined using Gene Imager 4.05 and the manufacturer's size standard of 50 to 350 base pairs. Individuals were randomized on all gels to avoid allele size scoring bias. This is the end of the microsatellite genotyping section. Genetic diversity. Mean observed and expected heterozygosities for all populations across all loci were calculated using Harlequin version 3.5. The inbreeding coefficient FIS was estimated using Harlequin version 3.5 and tested for deviations from 0 using a permutation test of 1,000 permutations with significance values adjusted using the Bonferroni correction for multiple tests. Inbreeding coefficients were significant for 8 of 32 populations. Therefore, we tested for the presence of null alleles using microchecker version 2.2.3. Null alleles were identified in 5 of 8 populations with significant FIS, so we used FRENA to estimate global and pairwise standard FST and unbiased corrected FST using the excluding null alleles or ENA method. ENA-corrected and uncorrected pairwise FST values were significantly different in a paired 2-sample t-test. However, both measures were very highly correlated, and the absolute mean of the differences in FST is unlikely to be biologically meaningful. We therefore used uncorrected FST values as the response variable in our isolation by distance models described below. We used bottleneck version 1.2.02 to test for recent bottlenecks or founder effects in the sampled populations, as bottlenecks could also lead to changes in allele frequencies and heterozygosity. Mean allelic richness, or A subscript R, for all populations at all loci was estimated using the Standardized package in R. We tested for an effect of colonization history on genetic diversity by regressing mean population allelic richness against the shortest water distance from the putative site of first introduction using a simple linear regression in R. The expectation is that sites close to the original introduction site have not undergone secondary bottlenecks, have had time to recover from founder effects, and may have received supplementary introduced individuals from other areas and thus should exhibit the highest genetic diversity. We also tested for significant differences in allelic richness among clusters identified in structure and NJ trees using an analysis of variance in R. This is the end of the genetic diversity section. Population genetic structure. Due to the inherently non-equilibrium nature of population structure in recently invaded species and our desire to characterize mechanisms of range and expansion, we first characterized large-scale population genetic structure using individual-based clustering. We used the program Structure 2.3.4 to perform genetic clustering of individuals with an admixture model, with location prior information included. All runs were performed for k equals 1 minus 11 with 10 replicates for each k and 250,000 burn-in iterations, followed by 750,000 MC iterations. The Ivano et al. In 2005 change in k method, calculated using a custom R script, was used to select the most likely k. Bar plots based on the most likely replicates were constructed using a custom script in R. We also tested for genetic structure among sampled populations using a population-based method by calculating genetic distances based on the Cavalli-Sforza chord distance using the genet.dist function in R package Heath, and then constructing a neighbor joining NJ tree based on these distances using the NJ function in R package ape version 3.5. Both structure and NJ tree identified 3 populations, Hastings, Midland, and Port Severn, as highly distinctive, so we also performed both analyses with these 3 populations excluded to assess their influence on results. This is the end of the population genetic structure section. Isolation by distance. We tested for an effect of geographic distance on genetic similarity between populations using the mantle.test function in R package ape version 3.5. Population site genetic divergence was assessed using FST calculated in FRENA described above, while geographic distance was measured as the shortest water distance between sites. Additionally, pairwise genetic distances were linearized using the FST divided by 1 minus FST conversion and were regressed against the shortest water distance between pairs of sites using a simple linear regression in R. Because samples were collected at a fine spatial scale in Lake Erie, we were able to test the hypothesis that populations in eastern Lake Erie were founded via ballast water transport from the original founding site near Lake St. Clair. We tested the relationship of genetic distance and geographic distance between populations in the St. Clair River, Detroit River corridor, and populations in eastern Lake Erie. In this case, we suggest that eastern Lake Erie populations were likely founded as the result of ships deballasting prior to entering the Welland Canal near Port Colborne, and subsequently spreading naturally westward. Thus, the predicted pattern should be one of increasing genetic distance with decreasing geographic distance to Lake St. Clair. This is the end of the isolation by distance section. Assignment and dispersal. To investigate individual dispersal among sites, we used the Bayesian genotype assignment approach outlined by Ranalla and Mountain in 1997 in the computer program GeneClass 2.0. To identify successful assignment, we estimated a likelihood ratio as the highest rank probability for assignment divided by the second highest rank probability. If the likelihood ratio obtained was greater than 4, meaning it was 4 times more likely to originate from the higher ranked population than the next highest ranked population, the individual was deemed successfully assigned. The likelihood ratio threshold of 4 is arbitrary, but sensitivity analyses showed that our results did not vary qualitatively with threshold values ranging from 2 to 9, though the numbers of successfully assigned individuals did change. Individuals were categorized into one of 3 categories: self-assignment, migrant of known source, or unidentified. We performed individual assignment initially with all 32 populations, and subsequently with populations grouped into the 6 clusters identified in the NJ and structure analyses. This is the end of the assignment and dispersal section. This is the end of the materials and methods. Results. Genetic diversity. Observed heterozygosity values ranged from 0.42 to 0.65, while expected heterozygosity ranged from 0.51 to 0.66. Estimated global FST was 0.067. Standardized allelic richness ranged from 3.5 to 6.8 at n equals 24, with the highest values recorded for St. Rose Beach and LaSalle near the putative original site of introduction in the St. Clair River, and the lowest value recorded at Port Severn in eastern Lake Huron. Mean standardized allelic richness also differed significantly between the three major population clusters identified in our structure analysis and neighbor-joining trees, with the highest mean recorded for the Western Lake Erie cluster, including McFarland and Trenton in eastern Lake Ontario, and similar means found for eastern Lake Erie and Lake Huron. Allelic richness was low overall for the three highly distinctive populations, Port Severn 3.5, Hastings 4.0, and Midland 5.1. Standardized allelic richness was negatively correlated with distance from the putative site of first introduction in Lake St. Clair. Three populations fell significantly further than expected from the regression line. Two of these populations, Trenton and McFarland, had anomalously high allelic richness for their distance from the St. Clair River site, while Port Severn had anomalously low allelic richness. Excluding these three populations and rerunning the linear regression improved both the significance and the fit to the regression line, but overall pattern remained similar. Bottleneck results identified two populations, Hastings and Port Severn, as potentially having undergone a recent reduction of their effective population size. This is the end of the genetic diversity section. Population genetic structure. The patterns of population divergence identified were generally consistent between DC and J trees in our structure analysis. The NJ tree identified three major clusters, approximately corresponding to 1, the western half of Lake Erie, 2, the eastern half of Lake Erie, and 3, Lake Huron. The best fitting or highest likelihood K equals 6 structure result showed a similar pattern, with three major clusters corresponding with the same groupings as in the NJ tree, and three additional clusters corresponding with highly distinct individual populations. These three populations, Hastings, Midland, and Port Severn, were highly divergent, characterized by unusually long neighbor-joining branch lengths in the phylogenetic tree, and each assigning to a unique cluster in our highest likelihood structure result. However, dropping these three populations and recalculating DC distances resulted in a tree that was essentially structurally unchanged, suggesting that these populations did not disproportionately affect the overall pattern. A few populations were assigned to genetic groupings in both analyses that did not correspond to their geographic locations. Three populations, Burlington in western Lake Ontario, Colchester in western Lake Erie, and Port Colborne in eastern Lake Erie, were genetically similar to populations from Lake Huron. One population, J. Gould, sampled in western Lake Erie, was genetically similar to populations in the eastern end of the lake. Finally, two populations from eastern Lake Ontario, McFarland and Trenton, were closely related to populations from western Lake Erie and the St. Clair River-Detroit River corridor. This is the end of the population genetic structure section. Isolation by distance. Overall, round goby population structure reflected a complex pattern of subdivision and isolation by distance in the eastern Great Lakes. There was strong support for the correlation of genetic and geographic distance when all populations were considered. However, closer inspection of the distribution of genetic distances shows that all of the largest pairwise genetic distances involve two of the three highly divergent populations identified in the NJ tree and structure analysis, Port Severn and Hastings. Mean pairwise FST divided by 1 minus FST plus or minus the standard deviation for Port Severn was 0.17 plus or minus 0.04, while for Hastings it was 0.21 plus or minus 0.04. Mean pairwise FST divided by 1 minus FST plus or minus the standard deviation for all other comparisons was only 0.05 plus or minus 0.03. Pairwise comparisons involving the two populations in eastern Lake Ontario, McFarland and Trenton, also had larger than expected residuals from the regression line. In this case, pairwise FST divided by 1 minus FST value between these two populations and populations in western Lake Erie fell well below where they were expected, based on geographic distance. Therefore, we also performed the linear regression analysis excluding the four putative outlier populations, Hastings, Port Severn, McFarland, and Trenton, as their genetic structure may be more consistent with long-distance anthropogenic transport than with natural or semi-natural stepping stone dispersal. With the four outlier populations excluded, there was still strong statistical support for the correlation of genetic and geographic distance. Pairwise comparisons between populations near the original introduction, St. Clair River, Lake St. Clair, Detroit River, and populations in eastern Lake Erie showed a significant pattern of decreasing population genetic distance with increasing distance, consistent with an initial founding event near Port Colborne and subsequent diffusive spread westward. This is the end of the isolation by distance section. Assignment and dispersal. Bayesian assignment analysis using all 32 populations demonstrated that we were able to successfully assign 662 of 1,958 individuals based on our conservative likelihood ratio threshold. 448 individuals were assigned to their population of origin, while 214 were identified as migrants of known origin. Of migrants of known origin, 127 were assigned within the same structure cluster, while 87 were assigned to a population in another structure cluster. Per population, successful assignments ranged from 8% in Amherstburg to 96.8% in Port Severn. The average percentage of successful assignments was 30.9% plus or minus 20.8 standard deviation across all populations. On average, more individuals were assigned to their population of origin, 19.5 plus or minus 24.1%, than were identified as migrants of known origin, 11.4 plus or minus 6.0%. The percentage of self-assigned fish ranged from 0% in McFarland, Northern Indiana, New York 3, and New York 6 to 95.2% in Port Severn, with the highest percentages seen in the three highly divergent populations, Port Severn, Hastings, and Midland. The percentage of fish identified as migrants ranged from 0% in Hastings and Burlington to 25.8% in Maitland River. Among the 214 migrants of known origin that were identified, the largest fraction of individuals, around 40%, moved less than 100 kilometers, though distances ranged from 5 to 867 kilometers. With populations grouped into the 6 clusters identified by NJ in structure analysis, we were able to assign 1,261 of 1,958 individuals. Of the individuals confidently assigned to one of the 6 clusters, 1,075 were assigned to their cluster of origin, while 186 were assigned to another cluster. This is the end of the assignment and dispersal section. This is the end of the results section. Discussion. Our results are consistent with the hypothesis that ballast water transport, both in the initial colonization and in secondary spread, has played a dominant role in the population genetic structure around gobies in the Great Lakes. The current population structure, which has been suggested to have been relatively stable for more than a decade, primarily resulted from the movement of large numbers of individuals via ballast water. Initially, these individuals were moved from the native range and secondarily within the Great Lakes. Further movement of individuals involving both natural migration and human-mediated transport of small numbers of individuals has served to establish some outlying populations and spread individuals out of the initial points of establishment. The dominance of ballast water transport in the round goby example highlights one major weakness of current ballast water management. Although requirements exist to prevent the introduction of non-indigenous species to the Great Lakes, once a species is in the Great Lakes, it can be quickly transported throughout the system, rendering any attempt at management too little, too late. Within the invaded range, we identified 3 main clusters in our neighbor joining trees, approximately corresponding with Lake Huron, Eastern Lake Erie, and Western Lake Erie/Lake St. Clair, plus 3 highly divergent populations associated with these lake clusters. The same pattern was identified in our structure analysis, with 6 clusters identified and closely matching our neighbor joining results. Previous authors have suggested that some of these areas, for example the Bay of Quinte in Lake Ontario, are likely the recipients of independent introductions from outside the system. We argue that there is no need to invoke multiple introductions to account for the observed genetic patterns. Instead, the pattern of population divergence is consistent with natural and human-mediated dispersal within the Great Lakes following an initial introduction in and around Lake St. Clair. Although multiple introductions from genetically divergent sources may have contributed to the current distribution of round gobies in the Great Lakes, we would argue that it is a relatively small part of the whole story. Secondary transportation events and range expansions within the Great Lakes likely played a more significant role. The anomalous similarity of geographically disjunct populations, including Trenton and McFarland in the Bay of Quinte, with the St. Clair River populations, Port Colborne, and the St. Clair Group and Burlington, and the populations in Lake Huron give evidence for a dominant role of ballast water transport, followed by secondary diffusive spread. Supporting our hypothesis is the fact that many of these anomalous sites are near ports or rivers and canals where ballasting and deballasting is likely to occur. Deballasting before entering narrow, shallow channels allows ships to increase trim and improves maneuverability and has previously been shown to be associated with sites of first discovery of non-indigenous species. Several studies on the dispersal of invasive species have identified human transport as critical to mediating dispersal and range expansion, including the round goby in the Great Lakes and Europe. In this study, genotype assignment analysis characterized round goby dispersal distances in the Great Lakes as highly variable, with distances ranging from no dispersal, individuals assigned to their source population, to over 800 kilometers, a distance clearly indicative of human-mediated dispersal if true. Here, long-distance dispersal mainly reflects ballast water dispersal plus the transport of small numbers of individuals via bait buckets or recreational boats. The dispersal events in the range of approximately 600 to 800 kilometers are migrants primarily moving between the lower Detroit River, western basin of Lake Erie corridor, and the Bay of Quinte in McFarland and Trenton, or from Burlington to Lake Huron via ballast water. The similarity of the Burlington sample and Lake Huron populations, along with assignment results that indicate movement of a number of individuals from Burlington to Lake Huron, suggest that Lake Huron populations may have been founded by individuals brought from the port at Burlington via ballast water. An alternative or additional route may have been overland transport, given that the distance between sites is relatively short. However, given the relatively high allelic richness in Lake Huron, our results are more consistent with ballast water than bait bucket transport. Unsampled areas in the south of Lake Huron would be expected to show a secondary contact zone between this Burlington Lake Huron cluster and the Lake St. Clair cluster, as natural dispersal filled in the gap between the founding sites of the two clusters. Other putative long-distance transport events are between the eastern Lake Erie cluster and Lake Huron, potentially consistent with bait bucket transport. Dispersal in the range of 100 to 400 kilometers primarily reflects migration between western and eastern basins of Lake Erie. Supporting our hypothesis that the region of Port Colborne is a secondary introduction site, several long-distance individual assignments are between this site and populations in the western Lake Erie cluster. Although incorrect assignments could occur, our identified dispersal pathways are consistent with FST values that reveal low genetic distance between sites in the St. Clair River western basin of Lake Erie corridor and the Bay of Quinte sites, as well as between sites in the St. Clair-Detroit River corridor and the extreme eastern basin of Lake Erie. The prevalence of significant pairwise FST values, coupled with local clustering within the structure analysis and J-tree and significant IBD relationships, suggest that natural dispersal within the Great Lakes was also a significant factor in overall colonization of the system. The significant correlation between distance from the St. Clair River, the first identified invasion site, and genetic diversity also supports this hypothesis, although it assumes that distance from the river is negatively correlated with time since introduction. Individual assignments also provided evidence for substantial natural dispersal. 39.7% of the identified migrants traveled less than 100 kilometers. Such limited dispersal distances are unlikely the result of ballast water movement. Although adult round gobies display high site fidelity, interactions between conspecifics may tend to push subdominant or juvenile individuals from areas of high population density into less populated regions. In flowing systems, such as the rivers connecting the Great Lakes, diel vertical movement of larvae may also play a role in transporting individuals between populations. While the vertical migration of round goby likely evolved as a predator avoidance or prey pursuit behavior, it would also serve to disperse larvae farther than expected for benthic, negatively buoyant organisms. The three populations that clustered separately in our structure analysis, Hastings, Midland, and Port Severn, located in the Trent-Severn waterway and far eastern Lake Huron, are strongly divergent from other populations in the dataset. Two of the populations, Hastings and Port Severn, showed evidence of bottleneck effects, and all three show significantly decreased allelic richness when compared to other populations. This suggests that they were founded by very small samples of individuals from elsewhere in the invaded range, and given the lack of large-scale shipping activity in the area, the presumptive mechanism of dispersal is bait bucket transfer. The spread of the round goby in the Great Lakes has been remarkably rapid. Since their initial appearance in 1990, the species colonized all 5 lakes within 5 years. This is surprising if we assume a dominant role for natural dispersal because, based on mark recapture studies, round gobies were characterized as highly phylopatric with limited dispersal tendencies. Thus, human-mediated dispersal clearly contributed to the rapid invasion of the Great Lakes by the round goby, as has also been shown in the Baltic Sea. We argue that ballast water transport has been dominant in structuring round goby populations in the Great Lakes. Moreover, we reiterate the problem of current ballast water management in the system. Although invasive species may be prevented from entering by current regulations, if they do manage to establish, they are likely to be spread rapidly throughout a system with few barriers. This is the end of the discussion section. This is the end of the recording of the journal article titled Human-mediated and natural dispersal of an invasive fish in the eastern Great Lakes.